Hello and welcome to General Biology at Rogers State University. And again, Dr. Martin. And this week we are covering Chapter 9 in your text, Sexual Reproduction and Meiosis. Now remember, in Chapter 8 last week, we talked about mitosis. And mitosis is cell reproduction, but it occurs in body cells, nerve cells, blood cells, muscle cells, bone cells, it's brain cells. It's reproduction where we start with a parent cell, divide into two daughter cells, and the genetic information is exact. In other words, the two daughter cells have the exact genetic information as the parent cell. Now that's chromosome number and genes and content. In other words, in mitosis, we create two clones. Remember, we said the most important thing that we have to do in mitosis is make sure those chromosomes make exact copies of each other. Well, that was mitosis. And remember, mitosis is used in asexual reproduction, primitive organisms, but it's also used in higher organisms like ourselves. It's used for growth and repair of injury, two key functions of mitosis. Well, now let's move on to sexual reproduction and what you might remember from high school called meiosis. It's a different process, still cell division, but a different process. Now, some organisms can reproduce asexually. We've mentioned this several times during the semester. It's where a parent cell like an amoeba, a paramecium, a euglena, a one-celled organism, usually fairly primitive, will bacteria do this too, where they will start with a parent cell and divide into two daughter cells, two identical offspring or two identical daughter cells. In this case, an amoeba reproduces asexually. There's no genetic exchange between two organisms, and that's what we call asexual reproduction. But it's pretty rare. One-celled organisms, bacteria, protozoans reproduce this way, but it's pretty rare. Most organisms reproduce sexually, and in sexual reproduction, it involves the use of sex cells, or what we call gametes. In humans, that's egg cells, sperm cells. In plants, it's pollen. In fungi, and other like mosses and ferns, they're called spores. But either way, there's genetic information that is exchanged between two organisms, and they contribute sexually. And there's an exchange of genetic information. Now, why do that? Sexual reproduction is expensive. Is it worth it? It's expensive because it exposes organisms to predation, to harmful impacts in the environment. A lot of energy is allotted to reproduction. Think of plants. A lot of energy is allotted to flowering and producing flowers. That's expensive. Uh, Organisms like frogs, fish, amphibians, and even lower organisms like jellyfish, they produce massive amounts of egg cells, sperm cells, and so that's expensive energetically. And so why do it? Well, this slide is a great description of why it is important. It's important because it produces genetic diversity. If an ecological catastrophe were to occur, genetically similar organisms would be much more susceptible than genetically diverse populations. And so it's all about genetic diversity. Much of your genome is contributed to diversity, genetic diversity, being different than the person next to you, being different than other organisms. That's genetic diversity. And sexual reproduction enhances genetic diversity. I think you, without having to force you to explain, I think you understand that when we just produce clones all the time, in mitosis, then every organism is very similar. But sexual reproduction, no organism is similar. Even you and a sibling, very different genetic genome. And so we've talked a little bit about that in the past. It's very important from a genetic diversity standpoint. So yes, genetic diversity is very important. So let's talk a little bit about meiosis. There are two kinds of cells when it comes to numbers of chromosomes. In humans, we have 46 chromosomes. In every cell of our body cells, what we call autosomes, and that's what we call a diploid number of chromosomes. You received a set of chromosomes from your mother, and you received a set of chromosomes from your father. You received 23 from each. When they came together in that first sperm cell, egg cell interaction, then we came together with 46 chromosomes. So a diploid number of chromosomes is two sets of chromosomes, one from your father, one from your mother. Those are what we call diploid cells. So in humans, we technically have 23 twin chromosomes, or what we call 23 homologous chromosomes. Homo means same. Remember that? Homo means same. Hetero means different. Homo means same. So we have 23 homologous chromosomes. This is a great picture of what we call a karyotype. And we can take your chromosomes from any cell of your body and create a karyotype. It shows all of the chromosomes that you have in your genome. And there are 23 pairs. Now my picture is kind of hiding it here, but there's a 23rd pair down here in the corner. If I were to change my picture here, you'd see the 23rd pair. I'll try to fix that here in just a minute. Well, no, here's the 23rd pair right here. So there are 22 pairs of chromosomes. There's 22, and then a 23rd pair here. Now these first 22 pairs are called autosomes. This last pair are the sex chromosomes. They determine your gender or your sex. Now, there's 46 chromosomes here. There's two number ones, two number twos, two number threes. Now, we just put them in order like this, so it's easier for us as scientists to study them. But there are you received 23 from your father, 23 from your mother, and they came together. And here they are, 23 chromosomes. There are 22 autosomes, and then one pair of sex chromosomes. Notice these two sex chromosomes are X chromosomes. So the karyotype of this individual is female. A karyotype of a male would be an X and a Y chromosome. Now, I need you to know something here. So here's two number ones. We know these are number ones because they are large. They also stain very similarly color-wise. So they're very large. The centromeres are right here. So these are large and they stain very similarly. Two number twos, two number threes. You received one of these from your mother, one from your father. Chromosome number 4, getting a little smaller. Did you notice that? You received one chromosome number 4 from your mother, one from your father. Here's chromosome number 5. Look at this. We get a little smaller. Our chromosomes get a little smaller as we go. Look down here. Chromosome number 19. Smaller, but they stain very similarly. These are chromosome 20, chromosome 21, chromosome 22. They stain very similarly. And so we call those a pair. One of those came from your mother, one came from your father. And so this is a karyotype. Karyotype is a graph that displays a person's chromosomes. And the first 22 pairs are autosomes. The last pair are sex chromosomes. We showed you on the previous slide, here's two X chromosomes. Here's an X and a Y chromosome. That's the gender of a male in humans. Now, other organisms, we begin to talk a little bit different. Notice the size discrepancy. in the X chromosome and the Y chromosome. The Y chromosome does not have very many genes on it at all. In other words, it doesn't code for very much in the male genome. You can probably, if you were to think about it, if I were to give you time, you'd think about what does the Y chromosome code for? Testosterone production, genitalia differences in male and female. So yeah, there are some things on the Y chromosome, but not very many things are coded on the Y chromosome. Many, many more genes on the X chromosome. You can imagine the larger the chromosome, the more genes on that chromosome, the more things that are coded for on that chromosome. In the mid 1990s, President Clinton started the Human Genome Project. And the Human Genome Project set out to identify every gene and its location on every chromosome in the human genome. It was a long project and highly enhanced by computers in the 1990s, but it began to give us a lot of information about genes. If we're going to do gene repair or gene therapy or gene manipulation, we had to know where every gene was on every chromosome. And so now we do know that. The human genome, one of the most scientifically advanced projects in modern history, was identifying the genes in the human genome and where they are at. And the Human Genome Project successfully did that about 25, 30 years ago. So we're still talking about these chromosomes. The two chromosomes of a certain pair. In other words, two chromosome number ones. You received one from your mother, the pink, one from your father, the blue. Each of these chromosomes have a lot of genes on them. We call these two chromosome number ones a homologous pair or twins. They are the same chromosomes. They have the same genes on them. By that I mean they code for the same things. For instance, hair color. You received one chromosome that had a gene on it for hair color from your mother, one chromosome that had a gene on it for hair color from your father. Those are homologous chromosomes, or specifically we call these identical alleles. Like this allele is a capital A, this allele is a capital A. In other words, this person received the same gene from the mother and from the father for a particular trait. Notice here in this gene, the gene is a little different. It still codes for the same trait. It's the same allele, but it's different. One is coded differently than the other. For instance, eye color. It's a little different. You receive different genes, maybe, from your father or your mother. Hair color, different genes from your father versus your mother. And so and down here, it's the same gene. So these are called alleles or genes. If some of the genes may be the same and some of the genes may be different, basically what we just said on the previous slide. Now, remember we said that in mitosis, the most important thing that happens before a cell can divide is we must make exact copies or replicas of every chromosome. Because you know what happens in mitosis. We have 46 chromosomes, and in S phase of interphase, we replicate every chromosome so that every chromosome has double the material. That's what's happened here. These are unreplicated chromosomes. These are replicated chromosomes. Notice they are held together by a centromere. Now you know what's going to happen in anaphase of mitosis. These two replicated chromatids separate and move towards opposite ends of the cell, and we end up with the same number of chromosomes in the two resulting daughter cells. And so these are called chromatids once they are replicated. And remember, We don't like mistakes in these. Well, this also happens in meiosis. But in meiosis, remember what happens here. We are resulting in sex cells. It's different. Mitosis was for growth and repair. But in meiosis, we are making sex cells. And sex cells are haploid. Remember, diploid says that there are two sets of chromosomes. Haploid says that there's one set of chromosome. Here in this case, this is an actual micrograph, a scanning electron micrograph of a sperm cell attaching to an egg cell. First of all, notice the discrepancy in size of these two sex cells. We're going to talk about that later. But here's a sperm cell with a flagella that attaches to an egg cell. These are gametes. They have 1/2 the genetic information of a normal body cell. So here's a sperm cell, egg cell, 23 chromosomes here, 23 chromosomes here. Those are haploid numbers. So when they fuse together in forming a zygote, now we're back to the diploid number of 46 chromosomes. I think you see that visually. I need you to kind of visualize that. We don't want diploid numbers in our sex cells. We need them to only deliver one set of chromosomes. In humans, 23. So that when fertilization occurs between a sperm cell and egg cell, we have each delivering 23 chromosomes. Now we're back to a diploid number of chromosomes, ultimately resulting in an infant or a human being. So zygotes, though, after fertilization has occurred, in this case, a sperm cell from a male frog impregnates an egg cell from a female frog, we have two sex cells joining together to form a zygote. After that, after fertilization, mitosis takes over, and then we begin to grow as an organism, embryonically, and then as an infant, or as a juvenile, or as a larva, or as a pupa, and eventually growing into an adult. So meiosis only results in sex cells. Once sex cells come together in fertilization, then mitosis takes over and the organism grows. So meiosis is essential in sexual reproduction. If we're going to reproduce sexually, we have to have gametes, haploid gametes. We call these germ cells because technically not all organisms that reproduce sexually necessarily have sperm cells. Some have pollen, some have spores. So we call these germ cells or gametes. They are haploid gametes that are essential in meiosis. The way this happens in meiosis, it's cell division. We are going from one cell to two cells, but we don't want it to occur just like in mitosis. In mitosis, we want the daughter cells to have the exact genetic information and number of chromosomes as the parent cell. Not so here in meiosis. In meiosis, we want to half the genetic chromosome number. So the way it happens in meiosis, because we're going to produce haploid gametes, is we replicate our chromosomes. Here they are, replicated. And then we divide it twice. Do you understand that? So we double our chromosome material and then split it twice so that we get to a haploid number. It's just simple math. We double our number of chromosomes or chromosome material and then split it twice so that we get haploid numbers of chromosomes. So we have what we call a meiosis 1, where the parent cell divides into two daughter cells. And then we have a meiosis 2, where each of those cells divide into two daughter cells. That's how we get to a haploid number for gametes, germ cells, or sperm and egg in humans. So here's what happens, and really fast. I'm not going to walk through this. We've already walked through all of these phases in mitosis, so I'm not going to do that again. But here's what happens in meiosis. We start with a diploid cell. In females, these are in your ovaries. In males, these are in your testes. We call these oocytes for egg cells, spermatocytes for sperm cells. It occurs in the testes of males, ovaries of females. We have a diploid number of chromosomes. We have 46 chromosomes here in this parent egg cell or parent sperm cell. We go through all the same phases of mitosis. Prophase, where the chromosomes are doubled but held together by a centromere. Metaphase, where they line up across the middle of the cell. Meta means middle. We have an anaphase, where these chromatids move apart from each other. These homologous twins move apart from each other. The cell splits in between them. Remember the figure 8 appearance here? And so now we have, at the end, we have two cells that have copies of twin or homologous chromosomes in them. We go through the same processes again. Now my picture is kind of covering up a fourth cell here. So this cell divides into two, this cell divides into two, but there's no replication that occurs in between there. So you see what happens. We start with 46 chromosomes. We double the material, divide it twice, once in meiosis 1, again in meiosis 2. We end up with 4 haploid cells, half the chromosome number in our gametes. So it reduces the number of chromosomes by 1/2. That's important, isn't it? Because this is going to result in a sex cell, not a body cell like in mitosis. These are resulting in sex cells, sperm cells or egg cells. Now, I'm not really going to walk through all of these phases. You know what happens in all of these phases. Again, there's a few differences between mitosis and meiosis. For instance, in mitosis and meiosis, we make a copy of all 46 chromosomes. They're held together. In mitosis, in anaphase, we split those two chromatids apart, and then we divide the cell in the middle. Here in meiosis, we have duplicated chromosomes, but the twins separate during anaphase, so that 23 twins go one way, 23 twins go the other. We split the cell in between, then we do it again. We split those chromatids so that we end up with 4. So we're not going to go through all of these phases, but same things happen in the phases of metaphase 1, of meiosis 1, where we start with a parent cell, end up with 2 daughter cells, and the cell splits in the middle in telophase, and then we go right into meiosis 2, where we have 2 daughter cells that just came from that first, and then we divide each of those so that we end up with 4 haploid daughter cells at the end of meiosis 2. Now I'm not going to walk through these phases. You know the general occurrences in each of these phases. So meiosis is important because it generates genetic variability. It generates genetic diversity. There are some things that occur here in meiosis that don't occur in mitosis. And this is one of those things we want to talk about. It's called crossing over. And this is crucial in ensuring diversity, in genetic diversity in organisms. In prophase 1, right here, these two twin chromosomes, they are doubled in material. Two twin chromosomes. Remember, you have one from your father in blue, one from your mother in pink. They line up next to each other in prophase 1. And look what happens. This arm of this chromosome crosses over the arm of this chromosome. Both of them are the same number 1 chromosomes or number 2 chromosomes or number 3 chromosomes, and they actually exchange genetic information. They kind of trade genes. And this is what we call crossing over. They literally cross over and then exchange genetic information. And it occurs here in prophase 1, the first meiosis 1. And so we call this crossing over. And in meiosis 2, it happens a little, it's not as important here in meiosis 2, but in meiosis 1, it's very important. Crossing over, it helps genetic diversity. It helps improving or enhancing genetic diversity in organisms. Now, we're going to stop right there, and we're going to pick up with some other information in the next lecture. We're going to talk a little bit about twins. We're going to talk a little bit about mutations and things like that. But that concludes our lecture on meiosis, or sexual reproduction.